Hello everyone! Today we will learn about fetal memory and placenta. For the embryo, fetal memory and placenta provide functions of protection, nourishment, breathing, and excretion, and they do not participate in the formation of embryo. Some structures also have endocrine function. After the fetus is delivered, the fetal memory and placenta are separated from the uterine wall and discharged out of the mother's body, generally called after birth. Fetal memory. Fetal memory includes chorion, amnion, yolk sac, allantoic sac, and umbilical cord. Look at this picture. You can see embryonic disc, amniotic sac, and yolk sac, and the green part is chorion. The chorion is composed of chorion plate, chorion stem, and chorionic villi. The trophoblast and extraembryonic mesoderm lining the inner surface of the trophoblast form the chorionic plate, based on which the various chorionic stems and villi are formed. After implantation, the trophoblast was divided into two layers, cytotrophoblast and syncytial trophoblast. This slide shows you the location of chorion and the chorion plate and villi. In this picture, you can see the embryo in the amnion, and the small ball-like structure is yolk sac, and the space outside of amnion is extraembryonic space. The dense filamentous structure is the villi, and many blood vessels can be seen in the extraembryonic mesoderm. The cytotrophoblast proliferated locally and extended into the syncytial trophoblast, forming many villus processes. In this way, the outer syncytial trophoblast and the inner cytotrophoblast form the primary stem villus. At the third week, the extraembryonic mesoderm extended into the stem villus, which was renamed as the secondary stem villus. After that, the mesenchyme of extraembryonic mesoderm in the stem villus differentiated into connective tissue and blood vessels, and connected with the blood vessels in the embryo. At this time, it was renamed as tertiary stem villus. In this slide, from left to right, you can see the primary stem villus, secondary stem villus, and tertiary stem villus. The trophoblast lacunae evolved into villus spaces between the stem villus, which was filled with maternal blood from the spiral uterine artery, and the villi were bathed in it. The embryo draws nutrients from the maternal blood through the villi and excretes metabolites. This animation displays the changing from the primary to secondary and tertiary stem villus. At the early stage of embryo, the villi distributed evenly on the whole chorionic surface. After that, due to the lack of blood supply, the villi of decidual capsularies gradually degenerated and disappeared, forming the smooth chorion without villi on the surface. The blood supply of the decidual bacillus is sufficient. The villi in this area are repeatedly branched and grow luxuriantly, which is called the villus chorion. It forms the placenta together with the basal decidua. The blood vessels in the villus chorion are connected with those in the embryo through the umbilical cord. After that, with the development and growth of embryo and expansion of amniotic cavity, amniotic memory, smooth chorion, and decidual envelope further protruded into the uterine cavity and finally fills with the decidual parentalis, and the uterine cavity disappeared. In the process of chorion development, if the blood vessels are not well developed or connected with the embryo blood vessels, the embryo may be stunted or die due to lack of nutrition. If the trophoblast cells proliferate excessively, the connective tissue in the villi becomes degenerated and edematous. The blood vessels disappear, the embryo development is blocked, and the villi are in the shape of grip or blister, which is called hydatid mole or cystic mole. If trophoblast cells become cancerous, they are called chorion carcinoma. Amnion. The amnion is a semi-transparent membrane which consists of a layer of amniotic epithelium and a small amount of extra-amniotic mesoderm without blood vessels. Amniotic cavity is filled with amniotic fluid, and embryo is immersed in amniotic fluid. The amnion initially adheres to the edge of the amniotic disc and is continuous with the ectoderm. With the formation of embryo, the expansion of amniotic cavity and the protruding of embryo into amniotic cavity, amnion wraps the body stalk to form umbilical cord in the ventre of embryo. In the expansion of amniotic cavity gradually makes amnion and chorion adhere to each other, and the extra amniotic body cavity disappears. The amniotic fluid in the early period of pregnancy was colorless and transparent, which was continuously secreted and absorbed by amnion. After the middle period of pregnancy, the fetus began to swallow amniotic fluid, and the excrement of its digestive system and urinary system and exfoliated epithelial cells also entered the amniotic fluid, which became turbid. With the growth of embryo, amniotic fluid also increased correspondingly, 1,000 to 1,500 milliliter in full-term delivery. In this picture, you can see the amnion is semi-transparent. The fetus is immersed in amniotic fluid. Functions of the amniotic fluid. The embryo, suspended by the umbilical cord, flows freely in the amniotic fluid. This buoyant medium permits symmetrical external growth of the embryo, prevents adherence of the amnion to the embryo, cushions the embryo against injuries by distributing impacts the mother may receive, helps control the embryo's body temperature by maintaining a relatively constant temperature, enables the fetus to move freely, thus aiding musculoskeletal development. Baby swimming is to let the baby to independent exercise in water, just like in the amniotic fluid, and use the gentle caress of water wave to promote the intellectual development and the healthy growth of the newborn. The yolk sac is located on the ventral side of the primitive digestive tract. A large amount of yolk is stored in the yolk sac of the embryo of birth, which provides the nutrition for the development of embryo. There is no yolk in the yolk sac of viviparous animal, including human embryo, and its appearance is also a phenomenon of phylogeny and evolution. At the 6th week, the yolk stalk closed and the yolk sac gradually degenerated. By the 9th week, the yolk sac has shrunk to a pear-shaped remnant about 5 mm in diameter. After the yolk sac of human embryo is wrapped in human umbilical cord, the part connected with the primitive digestive tract is relatively narrow, which is called yolk stem. It is connected with the midgut by the narrow yolk stalk. By 20th week, the yolk sac is very small. Thereafter, it's usually not visible. Functions of yolk sac. One is that hematopoietic stem cells occur in the wall of yolk sac. Another one is that Primordial germ cells appear in the wall of the yolk sac in third week and subsequently migrate to the developing sex glands or gonads. Here, they become the germ cells. The allantois is a cecum extending from the tail of the yolk sac to the pedicle of the body. It opens to the ventral side of the tail of the primitive digestive tract as the tail of the embryo is folded. When the bladder evolved from the latter, the allantois became a thin tube from the top of the bladder to the navel, called urachus. The urachus will close and become the ligamentum umbilical medianum. The allantoic artery and vein form in the extraembryonic mesoderm of the allantoic wall, later evolved into the umbilical artery and vein in the umbilical cord. The umbilical cord is a cord-like structure connecting the umbilical part of the embryo and the placenta. The cord is covered with amnion and contains mucinous connective tissue. In addition to the atretic yolk sac and urachus, there are also umbilical artery and vein in the connective tissue. The umbilical vessels connect the embryo vessels with the placental villus vessels. There are two umbilicus arteries because they are longer than the umbilical cord. They are spiraling. The umbilical artery carries the fetal blood to the placental villus and exchanges the material with the maternal blood in the villus space. There is only one umbilical vein, which will send the blood and has absorbed rich nutrients and oxygen back to the embryo. At birth, the cord is 30 to 90 cm long and 1 to 2 cm in diameter. If the umbilical cord is too short, it's easy to cause premature separation of placenta and excessive bleeding when the fetus is delivered. If the umbilical cord is too long, it's easy to wrap around the limbs or neck of the fetus, which may lead to local dysplasia or fetal asphyxia.